Are we just reviewing today?
Yeah, pretty much. I'll just review chapter 10 and 11 because we already reviewed chapter 9 on Tuesday. So I'll review chapter 10 and 11. I was going to do, like, I'll probably write out just a Punnett Square, problem or whatever, maybe just the homework one, just to make sure everyone is good with that. Cause I usually that's something that we usually do together and actually write out. But yeah, pretty much. And then I'll just make sure everyone knows what's what for the lab. I just have to record the demo, but I have your assignment up and everything like that. So I'll just introduce that. And so that's it. Unless you guys want to go over something else. We're good. All right, so I am recording. Okay. All right, so let me share my screen. And I guess I'll go over chapter 10, 11 homework and also the quiz, and that'll be our review. And like I said, for chapter 11, I'll probably just go over a Punnett Square or two. I may do a blood typing one just because that one's slightly different, and then go from there. All right, so first things first, before I start that review, just so I don't forget, for the last lab, so one that mitosis and meiosis lab assignment, that was due today. And I just want to just remind you about that too, in terms of what would be on the, the last lab exam. It would just be being able to identify the different stages of mitosis, right? From pictures like this. Remember, this is a whitefish blastula. I'll pull pictures that are very similar to these. And then, so that's for mitosis. And then also for meiosis, being able to identify the different stages of both meiosis 1 and meiosis 2. Okay, so that's basically, I mean, in terms of knowing what happens at the stages, mitosis and meiosis, that will be on that the lecture exam. But for the lab exam, it would just be identifying the stages from the pictures. Okay, so that's that first part of the lab exam. And then the second part of the lab exam will be the lab that's going to be due next week that I'm going to show you just right now real quick. Just a quick introduction, and then I'm going to post that video of the demo probably within an hour or so after we get off. I just have to record it. I sort of have everything ready. All right, so let me go back. So that's that. I just want to make sure I reminded you guys about that. And make sure this exam 3 review sheet, make sure that is submitted by the end of the day today. I think I extended the deadline until like 11:59 p.m. tonight. I will have your next exam 3, posted tomorrow. It may be posted tonight. If it is, I'll send an email out just to tell you guys when it's posted, but it'll definitely be up by tomorrow morning. All right, so for the lab assignment, so these are just extra, the handouts, and I'll sort of refer to them in the demo. So as long as, and so that's what that is sort of, there's some background information, which is also in this PowerPoint, which I'm going to sort of start off presenting. And then there's also just some additional handouts on the micropipettes. So I'll kind of show that to you during the demo, so you'll understand what's what then. In terms of the assignment, it's here. There is a Word document in the assignment, okay? So you want to have that out and ready to go as you're listening to the demo. You can pretty much, fill that out and complete that lab report as you're listening to the demo. And then, and again, I'll be explaining everything there. So it's just going to be a Word document that has questions, also has the protocol or the procedure for the DNA extraction I'll be doing. And you can actually do this at home if you really wanted to. And then there's questions that move through and go through all the different parts. Okay, so that's going to be your assignment to complete that for that lab. All right. So anyway, that's that for the lab. This is going to be due though on Tuesday, because then the lab exam will be available Tuesday to Thursday, and then remember the final exam, I'm going to make that available probably starting Thursday of next week. So it's important that the lab is in Tuesday. If there's any questions Tuesday during our meeting, I can review that. You'll get the lab exam done and then the final exam, okay? Because next week is our last week of classes. Does anyone have any questions about that? It all sort of makes sense, right? So we don't have much left. Okay, so that's that for lab. So like I said, the last thing you have to do for lab is watch the demo. There's also an electrophoresis video that I'll probably show during the demo too, but it's there if you just need to go back to that as well. I'll put that up, like I said, in the next, once we get off, in the next hour or something. And that's due Tuesday, okay? And then, so that's and then you'll have the lab exam for that. So for lab, it's this lab assignment and the lab exam. And then for lecture, we already went over chapter 9. You had a chapter 9 quiz that was due Tuesday. For today, chapter 10 and 11 homework was due. And then also there was a chapter 10, 11 quiz, which is right here. So I'll go over those. The exam 3 review sheet is due, like I said, by the end of the day today. And so again, that exam 3 will cover chapters 9, 10, and 11. Okay. Again, does anyone have any questions before I move on just to review chapters 10 and 11? All right. Okay, so let's just look at the homework assignment, I guess, first. And then I'll probably go over the homework. The chapter 11 homework, I'm going to, like I said, I'll probably write out an example. I just have to kind of change my screen to a whiteboard. So I may leave that until after we go over the quiz. Or I'll decide how I want to do that. But let's go over chapter 10 first. So a big chunk of chapter 10, right, went through the procedure. Well, not actually, I guess it wasn't really a big chunk, but a part of chapter 10 went over meiosis, right, and the stages of meiosis, right? So basically what's happening there. Also, a lot of chapter 10 was comparing mitosis and meiosis, right, in terms of the procedures. So important that we understand, you know, why meiosis happens, when it happens, right? And also the differences between mitosis and meiosis, both in terms of purpose, but also in terms of kind of what's happening at the stages. So I hope you guys sort of saw that there was a lot of similarities between the actual sort of stages of mitosis and meiosis, but in terms of purpose and where and when it's happening is totally different, okay? So that is kind of the main chunk, but also in chapter 10, we talked about, you know, chromosomal abnormalities, right? So that's the kind of the second part. So the first question here in your homework questions, right? It said describe the 2 main ways meiosis contributes to genetic variation. So again, something very different that's happening in meiosis versus mitosis. So to review that, I mean, the short answer to that, right, is crossing over, which happens during prophase 1, right, of meiosis. And then it's independent assortment. Okay, but I did say describe, so hopefully we provided a little more explanation than just naming those 2 things, but those are the 2 things. But it's important to understand why and how, or sort of what is happening during those 2 things. Okay, so let me just pull that up just to review, because this is super, super important. All right, so in terms of the crossing over, I told you, you know, it happens during prophase 1, okay. But again, the reason why it's so important is that it's going to increase genetic variation, okay? Super important, because we're talking about meiosis, in which you produce gametes, right? That meiosis is the production of sex cells. The only place it's happening in the human body is either in the ovaries or in the testes, okay? So obviously it's super important that all of those cells are genetically different, right? This is why you're not the same as your brother or sister, because there's tons and tons of genetic variation that's involved in meiosis. And again, one is crossing over and the other one is independent assortment, okay? So for crossing over, basically you just have the homologous pairs line up with one another, right? So again, during prophase 1, these homologous pairs line up with one another. And I told you during the recording, you don't need to know about the nucleoprotein lattice or anything like that. But basically the way they line up is that there are certain proteins that help provide a framework for them to line up exactly, right? So that if you're talking about, again, always red will denote the maternal, right, chromosome, and then blue the paternal, right? The one that came from your mother, one that came from your father. Now, again, also notice that these are duplicated chromosomes, right? They have 2 sister chromatids because they're duplicated before that whole process starts. And what you notice is that, again, these have to be lined up exactly in that gene A has to line up exactly on the maternal and the paternal, okay? And what happens is you have exchange of these DNA segments from the maternal, the red chromosomes, and the paternal, okay, chromosome, right? And so what you notice happens is that you end up with a sort of mixture of different variations of these chromosomes, these daughter. So this is showing you the daughter chromosomes, right? Once the 4 daughter cells are produced when you go through all of meiosis. Okay, so this doesn't just look like, or you don't have the same chromosome as you had as just that maternal copy. Now there's a little bit of a mixture here, right? So you have little c instead of where you had big C originally. Okay, I hope that makes sense. I don't know if anyone needs more explanation, please let me know. I mean, it is sort of, you know, complicated kind of process, or maybe something that's sort of hard to conceptualize. But again, this is going to lend to genetic variation. So you're not losing or deleting any genes, you're just changing that the form of it, right? Different alleles. The alleles are sort of getting, I guess you can think of them being shuffled around. Okay, so that's crossing over. Now the other process that increases genetic variation is independent assortment. And all this means is that depending on how the homologous pairs line up, right, during metaphase 1, they're then going to go to those corresponding daughter cells, right, during meiosis 1. Okay, so that's all that means. And again, remember, you know, this is simplifying it, and it's only showing you 3 pairs of chromosomes, right? In humans, we have, you know, 2 pairs of 23 chromosomes, right? So this is only just showing you 3, but it's illustrating the point where you can have all these different combinations, right? And so there was a little bit, I went through a little bit in terms of kind of numbers and how much this sort of variation is increased, right? Or how many different possible zygotes you can get, right? So, so this is basically showing you here 2 to the power of 23, just because of this independent assortment. But then it's squared, right? Because you're talking about the egg and the sperm cell coming together to form a zygote, right? So the independent assortment is going to occur when the gametes are formed, the eggs are formed, also when the sperm cells are formed, right? And then again, you're going to square that when the zygote forms. So that's where this number, this comes from, okay? So that's a ridiculously large number in terms of variation that is just caused by independent assortment, okay? And then if you have crossing over, which we talked about occurring only once, you can see how much bigger this gets, okay? And crossing over often happens, you know, many more times than once, all right? You don't need to memorize these numbers, just so you know, in case somebody's gonna ask that. That's not what it's about. It's just meant to illustrate how much variation this really brings about, okay? All right, so like I said, you know, the first part of this chapter, we talk about, you know, the purpose of meiosis, right? The genetic variation that's brought about during meiosis, the stages of meiosis, meiosis 1 and meiosis 2. Then the second part of the chapter talks about, you know, what happens if things go wrong, right? I also told you that there's a lot of comparison between meiosis and mitosis. Make sure you can write out some similarities and some differences between meiosis and mitosis. There'll definitely be an essay question that asks you that, okay? All right, so that's all that. And then, like I said, when we get, so there was also a section where we talked about sort of the cycle of life, right? Just understanding where meiosis and mitosis fits into the cycle of life. I think it's pretty easy to understand. But like I said, the second part then talked about what happens when things go wrong, okay? For the most part, things go right? A lot of the times. However, if things go wrong during meiosis, right, you can end up with more chromosomes or less chromosomes, right? And we call that aneuploidy. Let me just see, what was that question? Oh, it asked about inversion and translocation. Okay, so I'll get to that in a second. So I'll answer that question number 2. So this is talking about a change in number, meaning you have more chromosomes than you should or less chromosomes than you should, okay? And this is going to happen due to something called non-disjunction, okay? And so I sort of illustrated that here, showing you non-disjunction, meaning, you know, these chromosomes aren't separated the way they should. Okay, so meiosis does not occur normally. So you end up with some of these cells that have too little chromosomes and some that have too many. Okay, and there really are very few I guess, changes in chromosome number in terms of in your autosomes, right? Not in the sex chromosomes that lead to viable offspring, okay? So it says, in fact, it says here 3 autosomal trisomies are viable beyond birth. I think that that's still true. I don't know the, I mean, I don't know if we looked it up now, if there was some other, you know, autosomal trisomy that was discovered. It's possible, and it's possible that in the future there may be, but so right now, or when this book was printed, there were really only 3. So that means, you know, there's only, this happens a lot more, but those offspring or those embryos are not viable, right? So there is no disease associated with it. So the most popular, not most popular, the most common, or the, I guess the one that we, most identifiable is Down syndrome, okay? So that's trisomy of chromosome 21. So another thing to just point out, we should be familiar with looking at one of these karyotypes and know what this is, right? So if I showed you a karyotype like this and asked, you know, what's wrong with this, or does this look like a normal karyotype, we should be able to identify that no, it does not, because there are 3, right, copies of chromosome 21 where there should only be 2. Again, remember, if we talk about humans, we have 2 pairs of 23 chromosomes. One of those pairs being the sex chromosomes. All the rest of these here, right, 1 through 22 are the autosomes. Okay, and so you can see again, 1 maternal copy and 1 paternal copy. Okay, in this case, you have an extra of chromosome 21. All right, any questions, guys? Okay. We also talked a lot about changes in chromosome number that occur in the sex chromosomes, right? In the X or Y chromosome. Now, these are a lot more tolerable. So you actually can see with, you know, like Turner syndrome, it's a female, right? But instead of having 2 X chromosomes, just 1. Normal intelligence, and usually there's not many other things wrong except for some aberrations in sort of sex characteristics, okay? And then we talked about some other ones here, Klinefelter as well. Same thing, we should be able to identify what we're showing here based on these karyotypes, okay? Then the last part talked about changes in chromosome structure, okay? So these aren't, you know, 1 whole extra chromosome or 1 less chromosome, but part of the chromosome breaks off, right? And sometimes it may be reinserted and sometimes not. So the homework question asked about, I forget which ones now. Inversion and translocation. Okay, so if we look here at this picture, basically it's showing you some of these examples of changes in chromosome structure. Okay, in a deletion, there's usually it's the end, right? The end of a chromosome will just break off and you lose that altogether. Okay, duplication, you have an area of that chromosome that's duplicated. Okay, if you talk about inversion, you have a piece that breaks off, but then it inserts in upside down. So you have different, you change the order of the genes. Remember that these letters denote genes, right? So you think of it as gene A, gene B, gene C, okay? In translocation, you basically have these flip-flopped, right? So these ends both broke off and they sort of switch and get exchanged, okay? Any questions about that? All right, so again, we talked about some different diseases or disorders that are associated with changes in chromosome number and also changes in chromosome structure, okay? So 2 different things, different types of diseases. That's basically what this end of this chapter talked about. All right, there's a lot of information in these chapters, right? This is really dense, you know, biology. So if there's questions, please let me know. All right, so what I'm going to do is I'm going to move on to chapter 11. Oh, one other thing I just wanted to remind you of is that there are videos, right, or animations that are posted on Moodle. You'll see there's a folder that says animations, and I was just going to show you one just so you can kind of see what they're like. Some of them are helpful, like if you just need a little overview of, like, say, what's happening during meiosis 1.
Meiosis
Let's see if you can
after
hear.
interphase,
I'll check at the
in which the
recording, see if you hear
chromosomes
this.
of a diploid cell have replicated during the S phase. After the 2 successive cell divisions of meiosis,
I won't show
a
you another
single
one. I'll
cell
just show you
will
this one.
produce 4 cells, each having a haploid number of chromosomes. Prophase 1 of meiosis 1 begins with the condensation of chromosomes and the fragmentation of the nuclear envelope. Centrosomes, which duplicate at the beginning of meiosis, begin moving apart, and the spindle fibers begin forming. Early in prophase, homologous chromosomes align next to each other to form tetrads. A tetrad is composed of 4 sister chromatids. Crossing over will occasionally occur between the non-sister chromatids in a tetrad. During prometaphase 1, the spindle apparatus continues to form, and the spindle fibers attach to proteins at the centromeres of the tetrad. One pair of sister chromatids is connected by a spindle fiber to one pole, while the homologous pair of sister chromatids is connected to the opposite pole. The chromosomes align at the metaphase plate during metaphase 1. Notice that the tetrads are aligned along the metaphase plate rather than individual pairs of sister chromatids, as in mitosis. Anaphase 1 involves the separation or disjunction of homologous chromosomes, with each pair of sister chromatids moving to the opposite pole of the cell as the spindle fibers shorten. When the sister chromatids reach the poles of the cell, they detach from the spindle apparatus and the nuclear membrane begins to reform. A cleavage furrow forms, and cytokinesis results in 2 cells, each having a haploid number of chromosomes, with each chromosome consisting of 2 sister chromatids. This completes meiosis 1, and after a period of interkinesis, the 2 daughter cells start the second cell division cycle called meiosis 2.
All right, so I will not show you the meiosis 2 video, but that is there as well. So there's a bunch of animations that are posted there, and so sometimes for these, it's nice to kind of just watch that animation to go over the process. So just so you know, those are there. I just wanted to show you that. All right, so that's my review of chapter 10. Does anyone have any questions that pertain to chapter 10? No? Okay. So what I'm going to do is I'm going to just shift gears and I'm just going to review. I'm going to go over the homework questions for chapter 11. Like I said, that's really, I think the homework question is really just doing a Punnett square and going through that. So I'll let me pull that up. I don't think I ask you. I think that's the most of it. And then we'll review the quiz. But like I said, I just wanted to write out a Punnett square just to make sure we all understand that, and if we had a question we can ask. All right. Yeah. Okay. So for the homework here for chapter 11, right? So answer the following questions for a pea plant, right? Remember Mendel, all of his work was using the garden pea, and it was a very good model, I guess, organism for his experiments for many different reasons, and I go over that in the recording. But so this pea plant has a genotype of big T, little t, okay? And so we call that heterozygous, right? So we need to be familiar with all this genetic terminology that I go over in chapter 11. So like, if you don't listen to the recordings, you probably should definitely listen to these recordings for these chapters, because like I said, it's a lot, okay? All right, so heterozygote, right? So my first question says, what is the phenotype of this plant? Meaning, what do you see, right? When we talk about phenotype versus genotype. So the genotype is big T, little t, right? It's heterozygous. The phenotype is what you see. So in terms of that plant being tall or short, okay? So it's tall, right? Whenever you have the dominant allele, it's the plant, right, is going to express that dominant trait. Okay, so if we're talking about basic Mendelian genetics. Okay, so what's the dominant allele? I already said that, big T. What is the recessive allele? Little t, right? So the dominant allele is tall, the recessive allele is short. Okay, it said what offspring would be produced if this plant was crossed with another big T, little t plant. Okay, so let me switch my share so that I can sort of write out with my mouse. I can't guarantee that it's going to be very neat, But at least let me just work through this so you understand what I mean by being able to just draw out a Punnett square. And it will only just be a simple, single monohybrid cross. I'm not going to ask you to like write out any dihybrid crosses or anything like that. But just know that the dihybrid cross is really just looking at two traits simultaneously, and you're doing the exact same thing, except that you're doing it with two traits. All right, so there's definitely, I guess, you know what, let me just pull up the PowerPoint just for a second, just to kind of make sure I sort of hit on a couple different things. And then I'll show you that cross. I mean, it's in here as well. So I mentioned monohybrid cross, make sure we know what that is. The generations, right? Parental generation versus F1 versus F2. So the crosses that he did, right? The one that I asked you to kind of write out, and I know it's hard to show your work, I realized, as I wrote that, as I just look at that on Moodle, usually I have people write these out, right? So obviously it's easy to just show your work there. So hopefully you guys showed your work somehow. I probably should have made it a little bit easier to do that. But anyway, you could just kind of draw out a table, right? That would work electronically. So anyway, he did his crosses like this, right? The parental generation, this was a tall plant and a short plant here. And then he knows from the F1, you get heterozygotes. So what happens when you cross these two heterozygotes? So maybe I don't even need to write it out because I can show you here. That's the question I asked on the homework question, right? So you make your little Punnett square, you write the genotypes of each parent, right, on each side here, right? So this is showing you the male on this side and the female on this side. So big T, little t, big T, little t. And then you match them up, right? These 4 boxes represent the possible offspring that can be produced, okay? So again, here you're gonna have, the genotype's gonna be big T, right? A heterozygote, and then another, a homozygous recessive here, okay? So in terms of listing the possible genotypes, right? Because I will ask you this on the exam, tell me what the possible genotypes are. From this cross, right? Maybe something like this. So in this case, the possible genotypes would be, right, homozygous dominant, heterozygous, and a homozygous recessive, okay? And then I may ask you, in terms of, the ratio or the genotypic ratio, right? Or the frequency. So you should be able to figure out from this, it's 25% being homozygous dominant, right? 50% being heterozygous, because it's 2 out of 4, right? And then 25% chance of being homozygous recessive, all right? If you talk now about phenotype, okay, phenotype is what you see. So now this is whether it's tall versus short. So the phenotypic ratio is 3 tall to 1 short, or 75% tall and 25% short, okay? You need to be able to do all those things, and I'm not gonna give you the same exact example, right? I'm gonna ask you about something else. Or if I change it and I said, all right, let's do a cross where we have a heterozygous male, but we have a homozygous recessive female, you should be able to do the same thing, okay? Does anyone have questions about that? I mean, this may be something that you've done before, but sometimes I find that there's people that haven't done it before. So like I said, I won't use examples exactly like that. I could use any different example, right? For instance, this is unattached earlobes versus attached earlobes. I would give you all the information that you needed in terms of what is the dominant phenotype, right? And what's the recessive phenotype? So here, big E, the dominant allele, means unattached earlobes, little e, attached, okay? Now, again, remember that maybe it's sort of uninteresting to think about Mendel's pea plants, or even maybe it's also uninteresting to think about unattached or attached earlobes, but hopefully you find it interesting to think about genetic disorders, right? In terms of autosomal recessive disorders and autosomal dominant disorders. Remember, autosome just means any chromosome other than the sex chromosomes, okay? And so you could write out a pedigree, or a geneticist or a genetic counselor might write out a pedigree of a family if there is some genetic disorder that exists, right? To kind of figure out where it came from. And so anyway, you should be able to recognize these pedigrees, right? Understand, you know, again, if we see a yellow, if it's yellow shaded, that means that individual is affected. Okay, so in this case, you would be able to tell without me telling you that this is an autosomal recessive disease, because the only individuals that are affected are the ones that have a homozygous recessive genotype. Okay, if you're a heterozygote, you don't have the disease. We call them a carrier, right? If you're homozygous dominant, you also don't have the disease, because the normal allele is the dominant allele, right? If you talk about an autosomal dominant disorder, it's different though, right? Here, if you just have one copy of that dominant allele, you will have the disorder, okay? So again, be able to recognize the difference, or identify based on a pedigree that I show you, whether it's autosomal dominant versus autosomal recessive. And then I gave you a couple examples of some autosomal recessive diseases and autosomal dominant. There are others, but I just gave you a couple examples. And those were there. Okay, then the last part of the chapter, right? When you talk about genetics, you know, everything doesn't follow those simple, you know, laws that Mendel figured out, right? Because there are other sort of patterns of genetic inheritance that exist, right? More complex types of inheritance. And so that's what that last part of the chapter talks about. Okay, so make sure you're, again, familiar with that stuff as well. Listen to the recording. I go over it all together. One other thing, and again, I'll probably just do one of these. I'll stop my share and I'll write out on the whiteboard just one of these in terms of a Punnett square here for like blood type, okay? So I'll do that as the last thing, okay? So let me go over the chapter 10-11 quiz, and then I'll just do an example or two of a Punnett square, and so hopefully we can follow that along. Does anyone have any questions? Again, there's a lot of information in this chapter, so let me know if you have any questions. All right, so let me just go over the chapter 10-11 quiz. And then, like I said, I'll go through one problem there, and that's pretty much it. Then we'll be finished. Okay, so in terms of the, why is it not? I don't know why it's doing that. Start attempt. I don't know why it's showing it that way, but anyway, that's weird. Okay, blank refers to the total number of chromosomes which exists in 2 sets. Okay, so that's diploid. Remember, when we talk about diploid number, it's right, the maternal and the paternal copies, right? 2 sets, in our case, 2 sets of 23. Is a nuclear division that reduces, I don't know why I have it set for 2 questions per page, but whatever. Blank is the nuclear division that reduces the chromosome number from the diploid 2n number to the haploid number. Okay, so remember, this doesn't happen in mitosis. That only is going to happen in meiosis. So you're starting with a diploid cell, but you're ending up with haploid. Okay, because why? You have the sperm and the egg cells are haploid, so that when the zygote forms, you then have a diploid cell. All right, blank is the exchange of genetic material between non-sister chromatids during prophase 1 of meiosis. We went through this, right? This contributes to genetic variation that is crossing over, right? During blank of meiosis, the homologous chromosomes separate, okay? So this is the homologous pairs are separating, not the sister chromatids, right? And that's going to happen during anaphase 1, not anaphase 2. So anaphase 1, the pairs separate, and anaphase 2, the sister chromatids separate. During blank of meiosis, the sister chromatids separate, right? I just said that, and that is in anaphase 2. Blank produces 2 genetically identical diploid daughter cells. That's mitosis, right? Remember, you're just producing genetically identical cells during mitosis, but during meiosis, you're producing 4 haploid cells that are all genetically different, okay? The correct number of chromosomes in a species is known as what? euploidy. Down syndrome is caused by a trisomy of chromosome 21. There are human disorders that are caused by changes in chromosome structure, right? A blank is the presence of a chromosomal segment more than once in the same chromosome. Okay, so you can, that's called a duplication. All right, so here, now we have a monohybrid cross between a plant with a big T homozygous dominant genotype and a plant with little t genotype. What will be the genotypes of the offspring, right? So here's an example of like a question I might ask on the exam, and you may have to just write out that cross real quick, you know, with yourself. But basically, each one, you're going to end up with all heterozygotes, right? So I probably don't have to write that one out. A monohybrid cross was done between two heterozygous plants with round seeds, right? What are the phenotypes of the offspring? We just went through this, right? So guess I probably should have told you what was dominant and what was recessive. But anyway, the big R, I guess I gotta edit that question. I'll go back and look at everyone's quizzes if I need to fix that, right? Because I don't expect you guys to memorize what those phenotypes stand for, right? Or what the dominant phenotype is. I would always kind of tell you. But if you're talking about seed shape, right? Big R round is the dominant phenotype. Okay, so if we're talking about two heterozygotes, again, it's that 3-to-1 ratio, right? So it's 3 round-seeded plants to 1 wrinkle-seeded, okay? So no matter what trait you're talking about, you're always gonna get that ratio when you mate those two heterozygotes. All right, in the genotype, big E, little e for earlobe type. E is what? It's the dominant allele. Okay, again, that was one of the examples I gave you during the lecture. Mendel's law of blank states that each individual has 2 factors for each trait. Okay, so if you go back to his laws, right, we have those 2 laws, the law of segregation and the law of independent assortment. Okay, so if you, this is a crappy question too. I probably got to fix that question too. I feel like it should have been a little bit more elaborate, because where do we see, yeah, I mean, I guess it's the law of segregation, but I guess you can kind of see how, you know, well, these do all kind of fit together, but anyway. I probably should elaborate a little bit more on there, but if you look at the laws in the book, it sort of states what they say, but I'm not sure in my PowerPoint if I really state everything exactly as it's sort of outlined there. But so anyway, it is the law of segregation. All right, I'll probably, like I said, I'll go through and look at people's quizzes. If for some reason you guys got those 2 questions wrong, let me write a little note to myself. I'll fix that, but let me just edit that. See, this is why I like to go over it, because I realize sometimes that maybe my questions could be a little bit more clear. So what was that? That law of segregation. Sorry, I just want to write that down so I can make these a little bit more clear. And then the other one was the one about the round seeds. Okay. All right. I'll fix these 2 to just make it a little more. All right, in an autosomal blank disorder, the normal allele, right, capital A, is dominant, and only individuals with the alleles or the genotype, right, of homozygous recessive have the disorder. Okay, so this is an autosomal recessive disorder. Okay, so again, the recessive allele is the disease allele. All right, last one. Cystic fibrosis is an example of an autosomal recessive disorder. Okay, so remember I gave you a couple examples of autosomal recessive and autosomal dominant disorders. Okay, so like I said, I'll go through and kind of look at those 2 questions. I'll look at your guys' attempts, and if I have to edit the grades, I will, just because I feel like those could have been a little more clear, because as I was reading them, I was kind of like, huh? So anyway, any questions about that, guys? All right. Like I said, let me just stop this share real quick, and then I'm just gonna pull up my whiteboard real quick and just see if I can write out one of the problems neatly enough for you guys to see. And if not, I'll just abort that. But otherwise, I pretty much went through and reviewed everything from 10 and 11. And remember, I reviewed chapter 9 on Tuesday. The exam 3, which will be available by tomorrow morning, will cover chapters 9, 10, and 11. Okay. And remember, I'm going to also put up another lab demo. That lab is due next Tuesday. All right. What was I going to do here? Oh, I know. All right. So let me just, I guess I'll just show you one of these. Okay. Because these are different, but it still will show you the sort of Punnett square and how you do that and how you set that up. But it also then will illustrate something that's different than basic sort of Mendelian genetics, right? In this case, right, with blood type, it illustrates something called codominance, meaning one of these alleles doesn't mask the other. There really is no recessive allele, right? They're all dominant. So if you look at here at blood types, if you look at the genotypes here, right, if you have 2 big A's, you're blood type A. Also, if you only have one, A, you're blood type A. If you talk about blood type AB, right, you have one A allele and one B allele. So like I said, there's not one that's recessive and one that's dominant. That's why we call it codominance. They're equally expressed. Okay, if you're type O blood, you don't have A or the A or the B allele. All right. So let me, like I said, let me just unshare here. Let me just see which one I want to set up. All right, I'll do this one. Okay. All right, so let me do that real quick. We want to do share, and we'll do whiteboard, and we'll see how that works. Okay, so we're going to set up a cross. Like I said, it may not be very neat. Maybe I can do it. Okay, like this. So this is going to be, these are the genotypes of the individuals. Okay, so here's one. And then we're going to cross that with another individual that has a genotype of this. Okay, and a little I. No, it didn't let me do that. Anyway, all right, so then you would set up your set all this up the same way if we're talking about any, genetics problem, including those basic, well, excuse me, I don't know why it's saying my internet is unstable, but hopefully you guys can still hear me. All right, and then on this side, right, would be big I, A, and then nothing here, okay? So if we go into this square and we fill this out, we have big I, sorry, this is an A here. Okay, and then again, this one would be same thing, right? Over here now we have the B allele and the A allele, right? So A, okay. All right, and then down here we have I, A, the A allele, and then we have little I, right? And then over here, B, and little I. Okay, so you should be able to, so if I tell you here are the parents, you're going to cross these, you should be able to set this up. And again, I can basically do this for anything, for any gene or any trait. And remember, I went through this in the beginning of the review of chapter 11. We need to know the difference between genotypes and phenotypes. So here, these are the genotypes. These are the possible genotypes that you can get, right? So again, you're going to talk about, like 25% or 50% or 75% or 1 out of 4, 2 out of 4, however you want to word that, okay? But in this case, in terms of genotypes, right, you have a couple possible genotypes. You have this one, I big A, I big A. You have I big A, I big B, right? Big A and a little I, right? These are all the possible genotypes here, okay? In terms of phenotypes, right? What do we have? We have 25, well, we have 50% that would be blood type A, right? Because remember this, I'm not going to write them all out, but remember this genotype is blood type A, right? But also, so is this, right? So again, if I give you a question like this as an essay question, I'll ask you about genotypes, possible genotypes, and also possible phenotypes. So in this case, in terms of ratio or percentage of possible phenotypes, it is 50% that are type A blood. It's 25% type AB blood and 25% type B. Okay? So anyway, I just wanted to make sure I went through one of these because these were a little bit different. And again, you should be able to set up these Punnett squares the same way, doing just basic Mendelian genetics where we have a dominant allele and a recessive allele. And again, like I said, I'll always tell you what that dominant allele is. So on that quiz, I didn't tell you, right? Like I should have said big R is equal to round seeds. And so I'm going to edit that quiz to fix that. But on the exam, I'll always tell you that. So I don't expect you to memorize what the dominant trait is and what the recessive trait is. Okay. All right. So that's it, guys. Unless there are questions, I shall end it here. Again, the exam will be up by tomorrow morning. Yep. And we're good to go. All right, guys, let me know if there's any other questions. Otherwise, I'll see you guys again on Tuesday. So we have one more week left.
Hi, thank you.
Thanks, guys.